My name is Kyle Korsoven, a graduate student in the chemistry department here at the University of Michigan. I'm really interested in how proteins fold and misfold, and in the Ramon-Murthy lab I'm able to investigate just that. Looking at the amyloid beta peptide, commonly associated with Alzheimer's disease, I can look at how the protein misfolds and how that misfolding correlates to Alzheimer's disease pathogenesis. Specifically, I've been able to investigate how membrane bilayer systems found on the surface of cells affect the folding of the amyloid beta peptide, and I've used a variety of biophysical techniques to look at the structure of the early interactions between peptide and biological membranes. Additionally, I've been able to look at how very small molecule chaperones may be able to influence the amyloid aggregation of A beta, and how that aggregation can be tuned by structural modification of those small molecule species.
My name is Amit Pathadia. I'm currently a graduate student in the Department of Chemistry here at the University of Michigan. My work in the Ramon-Murthy lab focuses on studying HIAPP, or amylin, a common pathological feature in type 2 diabetes. Specifically, I use chemical and biophysical tools to study how natural product-based small molecules influence HIAPP aggregation, structure, toxicity. Furthermore, my work also focuses on understanding how membrane curvature and content influence HIAPP aggregation and how it's related to cellular toxicity.
My name is Kian Kamgar Parsi, and I'm an applied physics grad student in Rams's lab. And my project is focused on the role of micelle-like oligomers in the aggregation of a various number of different amyloids. So, micelle-like oligomers have been correlated with enhanced fibrillation and toxicity in both human IAPP and rat IAPP amyloids. And the focus of current studies is on examining the roles of these micelle-like oligomers in human calcitonin. So to do this, we study the micelles using both pyrene fluorimetry assays, as well as dynamic light scattering, circular dichroism spectroscopy, and also 1D proton and DOSY NMR. And we hope to elucidate the role that they play in aggregation kinetics, as well as potentially examining their role in toxicity.
Hi, my name is Michael McCullough. I'm a senior undergraduate student here studying biomolecular science. I'm an aspiring student of medicine. In the lab, I am working with the amyloid protein IAPP, which is associated with type 2 diabetes. And I'm studying the role of various membrane compositions and curvatures and the role it plays in the IPP aggregation process.
Hi, my name is Meng. I'm a fifth-year graduate student in the chemistry department of University of Michigan. I'm working on the cytochrome P450 project in the Ramon-Murthy lab. What we are particularly interested in are substrate binding to P450 and the electron transfer between P450 and its redox partners, cytochrome B5 and cytochrome P450 reductase. We mainly use solution and solid-state NMR technology to explore the structure and dynamics of protein-protein and protein-ligand complexes at the atomic level resolution. We also utilize other biophysics tools, including CD, DLS, fluorescence, ITC, TEM, and stopped flow spectroscopy to better characterize our system. During my PhD, I've been working on several projects, including one, investigation of the mechanism of how membrane affects the interaction between P450 and B5. Two, using nanodisk for membrane optimization to study complex formation. Three, to understand the mechanism of how substrates affects P450 B5 interaction. Four, to understand the function of the flexible linker of B5 and how it affects protein-protein interaction. Five, to solve the structure of the FMN binding domain of CPR, and then solving the structure of the complex between P450 and this domain, which helps in understanding the electron transfer pathway between the two proteins. Six, to understand the domain motion dynamics of CPR and how it is related to the interflavin and interprotein electron transfer.
Hi, my name is Katie Gentry, and I am a second-year graduate student in the program of biophysics. In the Ramon-Murthy lab, I am a member of the cytochrome team. We're looking at cytochrome P450. Cytochrome P450s are a family of enzymes that are ubiquitous, and they are in charge of the metabolism of many compounds. There's endogenous compounds such as vitamins and sterols, as well as exogenous compounds, including 70% of the drugs on the market today. In the cytochrome P450 catalytic cycle, there are 2 electron donation events that take place, one with cytochrome P450 reductase, but also with cytochrome B5. What I'm interested in studying is looking at the interactions between these 2 proteins or 3 proteins and using what's called a nanodisk as a membrane mimetic to study how a membrane can affect protein-protein interactions. Using various NMR technologies, we will be able to produce high-resolution structures of these complexes.
I'm Tirupati Ravala. So I'm currently a postdoc in Ram's lab. So I finished my PhD from Indian Institute of Science. Currently I'm in Ram's lab. I belong to a cytochrome team. So I'm working on studying the structure and function of membrane-bound proteins using nanodisk technology. Mainly my work involves the preparation of the nanodisk using different peptides and incorporate the membrane-bound proteins in the nanodisks and study its structure and function using NMR.
Hi, my name is Kamal Mroui and I am a research fellow in Professor Ramamurthy's group. I do work on bone NMR and we try to exploit bone properties using solid-state NMR spectroscopy. As you may know, bone is one of the most interesting but challenging materials in nature, and this is due to its complex heterogeneous structure. The 3 main components of bone are the organic part, which is mainly collagen, and the mineral part and water. Our primary tool for studying bone is magic angle spinning MES NMR spectroscopy, in which we use ultra-fast MES to look into the different components of bone and the interaction between the 3 components of bone, using, as I said, ultra-fast MES NMR.
Hello, I'm Nong Jun Zhang. I got my PhD degree last year and joined the Ramamurthy's lab. I'm currently a postdoc working on the methodology development of solid-state NMR under ultra-fast mass beyond 60 kHz. So in order to spin the rotor to be so fast, the diameter of the rotor is very small. So currently what we have is 1.2 millimeter rotor. We also had a collaboration with Dr. Yusuke Nishiyama in the gel company. So we had a good collaboration and did a great job in the development of solid-state NMR methodology. We also applied solid-state NMR to many different materials such as polymer, biomaterials, pharmaceutical compounds, and investigate the structures and dynamics of these different materials.